Hello, my name is Daryl Cole, and I'm a scientist in the bioanalytics team at Sartorius in the UK. Today, I will be talking about a poster that presented at the ISSCR 2024 entitled Optimizing Human Hepatic Organoid Development, Leveraging iPSC Differentiation and Advanced Analytical Platforms with Research Use Only Growth Factors and Cytokines. We'll begin with an introduction to iPSCs. So iPSCs, what are iPSCs? So iPSC is an acronym for Induced Pluripotent Stem Cells. So on this slide, I'll go over what stem cells are and their use cases in the clinic and in research in general. So these cells are quite unique in the fact that they have the ability to differentiate into multiple lineages depending on their type. So over on the right, we've got a nice little graphic here showing you the different types of stem cells. So we've got the totipotent, which can produce all of the cells within the human body, placenta and embryo included. And from that, we can derive pluripotent stem cells. And these are the stem cells that can then produce all of the somatic cells in the body, such as hepatic lineage, bone, neural, cardiac, and blood. They're a highly valuable research or clinical tool, as you can probably imagine, and they can be used to develop models of normal and diseased tissues from individual patients or from a pool of iPSCs that can be commercially sought. Let's get more specific and talk about iPSCs. So what are they? How are they derived? So these are stem cells that have been induced from normal tissues, such as fibroblast cells, and the expression of specific transcription factors are forced on these cells. This method was discovered in 2006 by Shinya Yamanaka and his team, and he used OCT4, SOX2, KLF4, and c-MYC as the transcription factors and forced expression in fibroblasts. The reprogramming can be done using a few different methods, such as Sendai virus, transfection, and episomal induction. And this is to get the transcription factors into the cells. And then over on the right, you can see a schematic which shows a simplified process for the creation of iPSCs from normal tissue. So the cells are harvested, and as mentioned previously, in this case, they're fibroblasts, and they're induced to transiently express the transcription factors that I've just talked about. From here, you can then maintain these cells in specific medium to create an iPSC line, and then choose to differentiate those iPSCs into numerous tissue types using specific media and growth conditions. And you can see a whole range of them here on the figure on the right. So now that we know what stem cells and iPSCs are, we'll just take a little look at the IncuCyte live cell analysis system in a bit more detail. Here is the IncuCyte live cell analysis system. On the right-hand side of this slide, you can see the system itself, so the little box, and you can see the tray that has been pulled out of the system. This is where all of the culture plates and flasks are placed. Usually you wouldn't see the system looking like this because it would be inside an incubator to maintain the right conditions for the cells that you'd be growing and monitoring. There's room for 6 plates and different flask formats. The smaller flasks you can see on the image there, you can get 3 of those per tray, but the actual 96 well, 6 well, those style of plates or larger flasks like T75s, you can fit 2 of those per tray, and you've got 3 trays on the actual slide itself. The benefits of the IncuCyte are that you can protect your cells. So when you're monitoring your cells, they're left undisturbed in the incubator, and they are maintained in the same position, so they're not moved around during the analysis, because the objective moves around beneath the cells as opposed to the cells and their plates being moved. You can set up your experiments and walk away and just leave the system to it. It's got an integrated image analysis software tool which allows users to view the experiments from the network connected to PCs. And because of all of this, you can improve productivity. As I said, you can accommodate multiple microplates in parallel within the system, and also multiple users and applications. And the main benefit really is that you can acquire and view images over time. So you can grow your cells, you can treat them with whatever it is you're treating them with, place them into the IncuSite, and then over time do real-time continuous image acquisition and analysis. And you get your images from that, but you can also produce short movies to show the changes over time, and you can analyze this data using the tools in the IncuSite system. Now we'll take a look at the IQ high throughput screening cytometer. This is the IQ high throughput screening cytometer. It's a high throughput, high content cell analysis solution. It can analyze both cells and beads for antibody screening and also functional profiling. So in the middle of the slide, you can see an image of the IQ there, and it comes with its own proprietary software called the Foresight software. And you can detect cell by cell, so thousands of cells per second, with multiple readouts for every single event. And you can analyze the natural conformation of cells and targets. Because of the design of the system, you can save precious samples. The sample sizes go down all the way down to 1 microliter per sample. So you can save any samples that you may want to use downstream. It's user-friendly as well, so there's no dead volume. The entire sample is taken to the detector. It's very, very fast, so you can answer more questions in less time. A 384 well plate is measured in around 15 minutes, and a 96 well plate can be measured in less than 5 minutes. Moving on, I'll talk about the iPSC differentiation workflow that's been used in the experiments where we differentiate iPSCs into hepatic lineage cells and also hepatic organoids. On this slide is a schematic of the hepatic differentiation protocol used to take iPSCs and transform them into mature hepatocytes and ultimately hepatic organoids. So we start with our iPSCs on day 0, as you can see on the schematic, and seed them into multiple wells in tissue culture plates coated with NutriCoat attachment solution. And this allows for collection and analysis at multiple time points throughout the workflow. Medium changes are performed daily, and this has to be adhered to during this protocol, with iPSC medium used until day 2, when the medium is exchanged for definitive endoderm medium, as shown in the schematic here. On day 4, a well of cells are collected for microanalysis, while the remaining cells are fed with hepatic endoderm medium daily until day 10. Another well of cells is then also collected on day 8 for microanalysis, and the medium is changed to the mature hepatocyte medium for the remainder of the time course. Cells are then collected at day 14 and day 25 for further microanalysis. And here, day 14 is also an important collection time point because the cells at this stage in their differentiation are ideal for development into hepatic organoids. So the cells are collected and then seeded into a 3D culture matrix such as Matrigel and fed with a specialized hepatic organoid medium until organoids begin to form. The organoids can then be monitored in the IncuSite and analyzed using the IQ as has been done throughout this differentiation process. So on the next couple of slides, I'll go through some of the results of this workflow. Here is the data collected while monitoring the differentiation of iPSCs into hepatic lineage cells. The micrograph on the left was taken using the IncuSite and displays the morphology of a normal iPSC colony at the beginning of the differentiation protocol. To the right of this image is a bar graph showing marker expression analysis performed on the IQ. As the data shows, iPSCs have a very distinctive morphology with individual cells packed into a colony. The marker expression shows a high level of pluripotent marker expression, approximately 90%, with minimal to no expression of non-pluripotent and hepatocyte markers. If we move on to day 4, there is a clear change in the morphology of the cells at the definitive endoderm stage. Cells have become much more spread out and have lost the colony-forming characteristics associated with iPSCs. This observation is also reflected in the marker expression, where we see a reduction to almost zero of the pluripotent marker, while non-pluripotent marker expression is elevated to almost 60%. Definitive endoderm marker CD184 is over 70% expressed, and CD99, a hepatic lineage marker, is over 80% expressed. By day 8, the hepatic endoderm stage, the cells have further diverged from their original iPSC morphology, becoming more confluent and spreading across the plate even more than on day 4. Marker analysis shows CD99 approaching 100% expression, while CD184 drops to below 60%, indicating a reduction of the definitive endoderm phenotype. Pluripotent marker expression is undetectable, while non-pluripotent marker expression is very low. Images from day 14, early mature hepatocytes, show cells with a high level of confluency, making discernment of individual structures difficult. However, it is clear that there are morphological differences compared to the previous time point, and of course the iPSCs. Marker expression profile is similar to the previous time point, day 8, with high levels of CD99 expression, around 97%, and around 60% CD184 expression. iPSC markers are low or not detectable. By the time we hit day 25, the last time point in the differentiation workflow, cells are at the late stage mature hepatocyte phase, and again the morphology has changed, but this time there are clear structures visible indicative of hepatocyte cells. Exhibiting large polygonal structure with round nuclei and prominent nucleoli, the cells feature hallmarks of mature hepatocytes. Expression analysis further confirms this, with cells showing approximately 50% non-pluripotent marker expression and negligible pluripotent marker expression. CD184 expression drops to the lowest it has been throughout the differentiation process at approximately 30%, while the CD99 expression remains high at 85%, indicative of a mature hepatocyte phenotype. Further characterization of cells throughout the differentiation workflow was also performed using the IQ. Intracellular hepatocyte markers albumin and HNF4A on the graph on the left-hand side in yellow and black respectively were analyzed using the time course. As the data shows, iPSCs have very little to no expression of either marker. But as the cells are differentiated towards the hepatic lineage, there is an increase in expression of both hepatocyte markers to a maximum expression level at day 25. This expression is higher than that found in the control liver-like cells, HepG2. To further characterize these cells, an endpoint cytochrome P450 enzyme assay was performed to analyze their functional activity. A CYP450 glow luminescence assay was used to compare CYP1A2 activity in iPSC-derived hepatocytes, iPSC control cells, and HepG2 liver-like control cells in the presence and absence of a CYP1A2 inhibitor, alpha-naptha-flavone. The graph on the right shows the data collected in this experiment. iPSCs had a negligible level of CYP1A2 enzyme activity, which was unaffected by treatment with the CYP1A2 enzyme inhibitor, while HepG2 cells had very high levels of enzyme activity that was significantly inhibited by alpha-naptha-flavone. iPSC-derived hepatocytes, although featuring lower enzyme activity than HepG2 cells, were more active than iPSCs, and their enzyme activity was significantly reduced upon treatment with the enzyme inhibitor, confirming the activity was specifically CYP1A2 enzyme activity associated with mature hepatic lineage cells. iPSC-derived hepatocytes were collected at day 14, as mentioned in the differentiation workflow, and were transferred to a 3D matrix to form hepatic organoids. On this slide, we have some representative bright field images that were acquired using the IncuCyte of the iPSC-derived hepatic organoids at multiple time points along their growth phase. So starting from the left of the slide, we have day 0 iPSC-derived hepatic organoids at the time of seeding. And as we move along from left to right, we see the increasing growth of the organoids with time. This initial data illustrates the 3D structure and growth of hepatic organoids derived from iPSCs using this differentiation workflow. And on the next slide, I'll go into a bit more of the analytical side of the IncuSight toolbox. Using the analytic capabilities of the IncuSight, a number of organoid metrics were measured over time during growth. IncuSight quantification analyses of organoid object count on the graph on the left show a steady increase in the number of organoids over time. Organoid object total bright field area, the teal plot, shows the measured area of organoids increasing over time, indicating stable organoid growth. The pink plot displays the organoid eccentricity over the time course, and as it is clear from the data, the organoids become less eccentric over time and become more uniform as they mature, and this indicates a healthy growth and development. And finally, to further characterize these organoids, an endpoint cytochrome P450 enzyme assay was performed to analyze their functional activity, as was done with the mature hepatic cells earlier. A CYP450 glow luminescence assay was used to compare CYP3A4 activity in iPSC-derived hepatic organoids, iPSC control cells, and HepG2 liver-like control cells in the presence and absence of a CYP3A4 inhibitor, verapamil hydrochloride. The graph on the right shows the data collected in this experiment, and the iPSCs had a very low level of CYP3A4 enzyme activity, which was unaffected by treatment with the enzyme inhibitor, as expected, while HepG2 cells had high levels of enzyme activity that was significantly inhibited by verapamil hydrochloride. iPSC-derived hepatic organoids, on the other hand, displayed the highest activity, and their enzyme activity was significantly reduced upon treatment with the enzyme inhibitor, indicating that these organoids were indeed of the hepatic lineage. iPSCs differentiated to hepatic organoids were further characterized by expression of surface markers using the IQ. So in the first graph on the left-hand side, we measured the pluripotency of the hepatic organoids by measuring SSEA1 expression for non-pluripotent and SSEA1 negative, SSEA4 trial 160 double positive populations, which were the pluripotent. And then hepatic differentiation was characterized by monitoring expression of CD184 and CD99 in a similar way to how the analysis was performed with the mature hepatic cells derived from the iPSCs. So what we can see here is that we have a low level of expression of the non-pluripotent markers, but no expression of the pluripotent markers, with a mid-level expression of CD184, just over 40%, and a high expression of CD99, which is indicative of mature hepatic lineage cell type. On the right-hand side, we also measured the expression of a LGR5 protein, which is a stem cell marker in organoids and 3D spheroids. And here what we found was that the iPSC-derived hepatic organoids had high levels of LGR5, which indicates a high level of proliferation, high level of proliferative capability of the organoids. So they were healthy and they were growing and very proliferative. Using the research use-only growth factors and cytokines in the Sartorius portfolio, iPSC-derived hepatic organoid growth was analysed using the IncuSight, as you can see on this slide. Organoids were grown in commercially available hepatic organoid media and an internally developed hepatic organoid media supplemented with RUO growth factors and cytokines, in addition to a basal medium without RUO supplementation. Micrographs show clear differences in growth across conditions, with organoids grown in basal medium appearing smaller and less developed, while the organoids grown with supplementation of RUO growth factors and cytokines are much larger, with the appearance of mature features within their structures. Commercial medium grown organoids are larger than the basal medium grown organoids, but they are also darker than the RUO supplemented organoids, indicative of increased depth within 3D structure. The image analysis graphs below confirm these observations, providing quantification of key metrics such as the number of organoids, the organoid total area, and the organoid darkness. For any further information, please see the following web pages. They have information on each of the machines that we've talked about today, and also an application note showcasing the hepatic differentiation that we've talked about today.